Hello Shishas, welcome to my lectures on why do we fall ill. Let us begin the topic with a quote as usual: an ounce of prevention is worth a pound of cure. As we're going to learn about the medical prospect, any doctor, they are very much familiarized with the terms that you study, but very deeper than these terms. And we also discuss most of the terms in the class, so I'm not giving any detailed information about this. Epidemic, endemic, pandemic, interferon, protein produced by infected viruses, inoculation, vaccination, incubation period, symptoms. Pathogen causes disease, parasite which takes shelter on host, and host which is present on the living body, infestation, vector, carrier, reservoir. So these three terms are most confused terms, so be clear with the definitions. And when you try to study the definition, have one example to understand it. Basics of immunology. So immunology is a study of resistance, how your body defends any pathogen that enters. And you can see the gentleman at your right, Robert Koch, who proposed germ theory. So he was the one who tried to propose scientific method in understanding the pathogens. And causes of diseases. There are many causes of diseases, including biological agents, like all living organism, nutrient agents that are carbohydrates, fat deficiency or vitamin deficiency, and chemical agents due to exposure of harmful chemicals, and physical agents like the internal factors, pH, salinity, and acidity. Mechanical agents, when someone gets injury in accident, it is considered as mechanical agent. And then the types of immunology or types of immunity. We have two types of immunity based on the origin: the innate immunity or non-specific immunity. These are naturally given by your parents. So anatomical, physiological, phagocytic, and inflammatory barriers, these are considered as first and second line of defenses. And acquired immunity, the antigenic specificity, diversity, and immunologic memory, self and non-self recognition, are considered as the third line of defense. The cells of immunity. The cells of the immunity are very active enough, they will be produced. So these are the names based on the various structures: helper T cells, inducer T cells, cytotoxic T cells, suppressor T cells, B cell, plasma cell, mast cell, monocyte, and macrophage, natural killer cells. See, antigens, antibodies, these are two antonyms you'll find in immunology. Antigen is a part of pathogen, and antibodies are part of our immune system, and immunoglobulins are Y-shaped structures you can see here, and this illustration shows antigens in different color and antibody. You can see a string of a rod kind of structure in Y shape. Now we'll go with an animation to understand more about these cells and its functions. So pathways of defense, you can see the biological causages, viruses, parasites, and bacteria. Now the physical barriers and innate immunity and adaptive immunity or antigenic specific immunity. The physical barriers, skin cells like skin, and innate immunity like macrophages, phagocytic cells that engulfs in antigens, and adaptive immunity. You can see the different cells that we just understood. This will be described. The physical barriers. So when pathogen enters, these three barriers will purely screen them, and this is an incident. So when we try to inject any bacteria or a rust metal exposed through a tissue, you can see pathogen entered through that, and our body responds here. You can see macrophages coming out, and they're trying to engulf them through phagocytosis. This is one situation, and what happens if this process fails and pathogens deeply enter inside your blood vessels and they want to uh, take more advantage of the less powered immune system. You can see the B cell, antigenic specific receptor, and antigen. So you can see antigen and pathogen and specific receptors attached to that. Okay, this is where antigens are processed. Now you can see how it's being tagged. Now helper T cells will be going to attach them. Interleukins will be transferred from this cell to other cell where it causes to kill the cell due to more amount of antibodies inside them, and also many antibodies will be duplicated to defend many other cells are produced. You can see many similar cells there, so all of them will be neutralized. Now it is one more situation. So when it is crosses the bloodstream, so the liver, inside the liver, now the cytotoxic T cell has antigen T cell receptor, so it is going to be attached. Then perforin proteins will be attached on its surface to tag them that they are infected. You can see swelling of the cell and bursting of the cell. So cytotoxic T cells will be helpful in this way to protect us from various diseases. We'll continue with the lesson. So we classified the diseases based on how they cause into two major types. So congenital diseases, congenital, that is came through came by birth by your parents genes, and next is the acquired diseases. The acquired diseases uh, happened due to exposing of some germs. So while you play in the ground, or due to infected guy in your class, like that. And acquired diseases will be given more priority here to understand them. So acquired diseases are again classified into two major types. One is communicable disease that can easily transfer to a healthy person. Non-communicable disease which will not be transferred to your friend or the healthy person. Next is methods of spread of communicable diseases. So direct transmission. So these direct contacts, droplet infection, contact with soil where pathogen survives, an animal bite. These are direct transmissions. Indirect transmissions by vectors, houseflies, mosquitoes, and cockroaches, and airborne, object-borne, waterborne, are few ways of indirect transmission. Let us continue with the causatives. First of all, you have to understand the causative uh, structure. So I have provided a general structure of the bacteria. You can see how it is how it has various structures like pili, gelatinous capsules, cell wall. So it is very microscopic in nature. You can see 1 micrometer Yeah, and based on how they actually form the groups or colonies or its shape, we classified rod-shaped, bacilli, spherical, cocci, spiral, etc. And the mode of reproduction. The mode of reproduction is really bad on the host, so it reproduces through binary fission. So the life cycle of it is very short, but the amount of the new population is really high, so that it is very difficult for the immune system to respond fastly and also to take control over them if a person does not have a strong enough immune system. So these are the bacterial diseases we're going to learn next few minutes: tuberculosis, cholera, uh, typhoid, diarrhea, diphtheria, leprosy. Tuberculosis, caused by the pathogen called Mycobacterium tuberculosis, mode of transmission, airborne, and then the incubation period 2 to 10 weeks. It is particularly produced, the symptoms caused by the substance called tuberculin. And symptoms: persistent fever, cough, chest pain, general weakness. And the diagnostic test is used, Mantoux test. And then the treatment is DOTS, directly observed treatment short course. So let us have a look at the animation. Then we continue with the next part of the presentation.
An estimated one-third of the world's population is infected with tuberculosis. One-third. And here's the culprit: the minuscule bacterium called Mycobacterium tuberculosis. But does that mean 2.3 billion people are sick? Fortunately not. Only a small proportion of them actually become sick. To understand why, let's take a closer look at the bacterium itself. When someone with TB coughs, they expel little drops of saliva that fly into the air. Some of them may contain the TB mycobacteria, which can then be inhaled by someone close by. The mycobacteria then continue their journey down the respiratory tract and into the lungs. That person is then infected with TB. And this is where it all becomes a little more complicated. The body detects the invasion and sends out its first line of defense. This includes special cells called macrophages, which engulf the mycobacteria and transport them to the lymph nodes, where they are neutralized. If they're neutralized, the person will have what is called latent tuberculosis. This means they're infected with the bacterium, but they don't have active TB disease, and they're not sick. However, in 5% of cases, the immune system is overwhelmed. The TB mycobacteria multiply and escape, and the person falls ill. This often happens with young children, whose immune systems are not fully developed. In other cases, latent TB can wake up and become active TB. This can happen months later, or even years later, especially in adults with weak immune systems. For example, people who are HIV positive, or who've had organ transplants, or who are undergoing chemotherapy. Once in the lungs, the bacteria may spread via the bloodstream or the lymphatic system to other parts of the body, to the lymph nodes, bones and joints, the digestive system, the central nervous system, the liver, or the heart. So what happens when someone has active TB? Let's imagine the infections in the lungs, as this is the most commonplace. The patient starts coughing, has difficulty breathing, and suffers from chest pains. If these symptoms last for more than a few weeks and are accompanied by fever, night sweats, and weight loss, the doctor has good reason to suspect tuberculosis. The next stage is diagnosis, and then treatment. The battle is far from over.
Nice video about tuberculosis. Now let us continue with the next disease, cholera. So cholera is caused by a pathogen called Vibrio cholera. You can see the structure. And the mode of transmission through contaminated water. Symptoms: diarrhea, dehydration, vomiting are more considered symptoms. And the incubation period lasts from few hours to days. Next is typhoid. So typhoid is caused by Salmonella typhi, and the mode of transmission through contaminated food and water. And the incubation period may maybe be around 1 to 3 weeks, and the symptoms are continuous fever, headache, and you can see a picture illustrating the symptoms: the skin rash, fever, abdominal pain, and the muscle aches. And the diagnostic test used is viral test in the beginning. And now let us look at diarrhea. Diarrhea is not considered as a disease, it is an intermediary dysfunction of the large intestine as well. You can see how large intestine will work to absorb the essential nutrients, and so when it fails, the stools are like flowing with the water when the reabsorption is completely failed. Anyhow, even it is considered as the disease when certain viruses are infected our system, and food intolerance and emotional distress and bacterial infection as well. And then uh, let us look at the diphtheria. So this is not part of our module, but somehow it is good to know them. So diphtheria is a pathogen caused by Corynebacterium diphtheriae. So you can see a boy with the symptoms illustrated. The lymph glands are swelled, and also this causes the boy to face difficulty in swallowing, sore throat, muscle weaknesses are some of the symptoms. And the vaccination is given in the form of combination DPT, diphtheria, pertussis, and tetanus. Next is leprosy. Leprosy caused by Mycobacterium leprae. Remember the similar group from TB. Modes of transmission. So it is transmitted through prolonged contact and nasal secretions, and symptoms of patches on skin, ulcer nodules, and the incubation period lies from 1 to 5 years. You can see the microscopic version of Mycobacterium leprae here. And then let us look at the next causative of disease, virus. So viruses are the non-living things outside the system, and when they enter into inside a living system, they start replicating, and they damage as much as they can to the host. And the viruses are broadly two shapes, so enveloped ones and unenveloped ones. And you can see the general structure: the surface protein, a viral envelope, and the genome inside presented. Sometimes, basically the viruses will have RNA as a chief genetic material, and even sometimes it might have the DNA. And you can see the single-stranded SS DNA or RNA, or DS double-stranded DNA or RNA. And you can see the scale nearly 0.01 microscopic meters. And influenza, hepatitis, rabies, AIDS are covered in the module, so we are going to understand them. Influenza is caused by the organism influenza virus. An incubation period is nearly 1 to 4 days. The transmission is through a droplet and sneeze of the person. So, generally influenza is considered as the fever, muscle aches, very high, and you can also see the chills, lack of energy, nausea, vomiting as other symptoms, and therapy is through the antiviral drugs. And then next is hepatitis. Hepatitis is caused by hepatitis A virus, and humans are main reservoirs for this. It transmitted through person to person by fecal oral route, like food consumed with the virus. An incubation period is commonly 28 to 30 days. It may be even around 15 to 50 days. So general facts about hepatitis: you can see how the liver got swelled, and it's going to cause nausea and fatigueness a lot. And then next is rabies. So rabies is caused by rabies virus, belongs to a a group called rhabdovirus, and here you can see the structure of rabies virus in the form of bullet shape, and you can see the lipid envelope. In the beginning, we just learned very few organisms have lipid envelope and matrix proteins, and ribonucleoprotein, and RNA. So incubation period for this is 1 to 3 days, and rabies is also called as hydrophobia. General facts about rabies. There are many facts you have to understand. So the clinical findings are like two findings are there. One is classic encephalitic and paralytic or dumb rabies. So both of them are very progressive, and even sometimes they may lead to death as well. And majority of cases, encephalitic rabies with hydrophobia and hyperexcitability, or even it is called as mad rabies. And you can see how the virus can pass through the central nervous system. If it is, the time of the symptoms, it takes how close it is to central nervous system. And next is you can see the various animals that can spread the disease. And then we look at AIDS. So AIDS is a disorder to immune system itself. That's why it is called as human immunodeficiency virus. The pathogen and diagnostic test is ELISA, enzyme-linked immunosorbent assay, very important in every exam you almost appear in your future. And incubation period is around 1 to 15 years, but even the patient may not get uh, positive in the diagnostic test very fast, very quickly. So it takes some time to the appear in the blood as well. So that's why it is actually takes a bit time. And next is therapy. So therapy is combination by antiretroviral viruses, and we have a special therapy called highly active antiretroviral therapy. So you might know recently the Nobel Prize is awarded for the for a scientist from Japan who has done work on autophagy, so self-killing of cells. So even those studies are very helpful to understand to understand the uh, to understand cure for AIDS. So let us have a look at animation about AIDS and continue with the next part of lesson.
When HIV first enters the human bloodstream, the virus circulates throughout the body. The protective surface of HIV is studded with particular proteins called GP120, and inside is a capsid that contains the viral RNA and viral enzymes. Initially, the GP120 protein on the virus is only able to bind to a cell surface protein on the macrophage called the CD4 receptor. A second receptor protein, CCR5, must also be present on the macrophage for the virus to enter the cell by endocytosis. Viral RNA and enzymes are released into the cell's cytoplasm. Reverse transcriptase uses the viral RNA to synthesize first a strand of viral DNA and then the complementary DNA strand. The viral DNA then moves to the nucleus of the cell, where it integrates into the host cell's DNA. Transcription of the DNA within the nucleus now results in the production of viral RNA. This viral RNA can serve as the genome for new viruses and can be used as messenger RNA to produce viral proteins during translation by the ribosomes. Complete viruses are assembled and released from the cell via exocytosis, which causes little harm to the macrophage. HIV cycles through macrophages over a period of years and continues to multiply while doing little apparent damage to the body. Eventually, the gene that codes for the GP120 protein is altered by mutation. The altered GP120 protein changes its co-receptor allegiance and now binds to a different co-receptor, CXCR4, which is found on the surface of CD4 plus T cells. The same processes occur within the T cells, resulting in new virus particles. However, as these new viruses leave the T cell, they rupture the plasma membrane, which kills the cell. As these newly released viruses invade and destroy other T cells, the body's immune response is weakened, and this leads directly to the onset of AIDS.
Now we'll continue with the next slide. So next causative is protozoans. The protozoans are in different forms. You can observe here. So all of this will involve in will have different modes of nutrition. So you can see amoeba, entamoeba, trypanosome, euglena, paramecium. The diseases we're going to learn is actually a module only consists of malaria, amoebiasis, color ulcers, sleeping sickness. We are doing additional. So malaria is a disease caused by Plasmodium species. The four uh, possible pathogens are Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium. So interesting thing is the Plasmodium is grouped under what we call as the sporophyte because their asexual mode is through only spores, okay, inside the blood cells or inside the host. Now we can see a patient has the symptoms illustrated here, and now we'll look at the historical event that took place to understand malaria. So these two gentlemen played a major role in understanding malaria and its life cycle. So incubation period is nearly 9 to 14 days, carried by female Anopheles mosquito. And you know about Lavrin, who discovered malarial parasite. That means he found that the plasmodium is present inside the body of unhealthy person, and you can see Sir Ronald Ross on your right, so where he discovered the pathogen transfer from unhealthy to healthy persons through female Anopheles mosquito. And now you can see the life cycle here. The begins with when the mosquito have the pathogen inside, it enters inside, and it starts going to the liver cell. In liver cell, it forms these sporozoids by bursting the cells, and then the RBC will be infected by the merozoids produced by multiple fission, and then most of them will go and infect all other RBC cells, and some of them will become differentiated, which we call as merozoites, and they will become gametocytes with male and female ones, and they will be present only outermost layer on the outermost layer as they flow in the blood. And when the healthy mosquito bites it again, so again it starts reproducing inside the stomach of mosquito through the sexual mode of reproduction, and again these sporozoids will come outside at the salivary gland and wait to infect the healthy person. So to understand in the well manner, let us go through the animation again.
When a female Anopheles mosquito penetrates human skin to obtain a blood meal, it injects saliva mixed with an anticoagulant. If the mosquito is infected with Plasmodium, it will also inject elongated sporozoites, motile spindle-shaped asexual cells, into the bloodstream of its victim. The sporozoites travel to the liver, where they enter liver cells and rapidly divide asexually. This asexual division, which is called schizogony, generates the next life cycle form, called merozoites. The released merozoites invade other liver cells and enter the host's bloodstream, where they invade erythrocytes. Once inside the erythrocyte, the merozoite begins to enlarge as a uninucleate cell termed a ring trophozoite. The trophozoite's nucleus then divides asexually to produce a schizont which contains several nuclei. The schizont then divides and produces mononucleated merozoites. The erythrocyte ruptures and releases toxins throughout the body of the host, bringing about the well-known cycle of fever and chills that is characteristic of malaria. Plasmodium enters a sexual phase when some merozoites in the erythrocytes develop into gametocytes, cells capable of producing both male and female gametes. Erythrocytes containing gametocytes do not rupture. Gametocytes are incapable of producing gametes within their human hosts and do so only when they are extracted from an infected human host by a mosquito. Within the gut of the mosquito, the gametocytes form male and female gametes. The resultant diploid zygotes develop within the mosquito's intestinal walls and ultimately differentiate into oocysts. Within the oocysts, repeated mitotic divisions take place, producing large numbers of sporozoites. These sporozoites migrate to the salivary glands of the mosquito, and from there are injected by the mosquito into the bloodstream of a human, thus starting the life cycle of the parasite again.
Nice, interesting animation. Now we learn about the amoebiasis. Amoebiasis is caused by a pathogen called Entamoeba histolytica. It happens to if contaminated food and water with this pathogen. And symptoms of it is see formation of ulcers. You can see it is its favorite spot to replicate, and even it causes bleeding. That's where we can be able to diagnose this. And next is Kalazer, caused by Leishmanian parasite. So generally the regions of infection is pain in the abdomen and even the fever. So it is very common to see swollen spleen, lymph nodes, and bone marrow. And that is the end of all communicable diseases. The non-communicable diseases. Non-communicable diseases are majorly divided into three major types. I want you to go through your book and try to learn more about it. So protein deficiency diseases, or even it is called as energy malnutrition. So kwashiorkor, marasmus are two. So kwashiorkor is curable, and marasmus is where extreme stage of the kwashiorkor. And mineral deficiency diseases, so due to lack of minerals inside body, anemia, goiter. So anemia due to less iron availability, goiter due to uh, iodine in salt. Generally, we take through salt. And vitamin deficiency, xerophthalmia, rickets, beriberi, B1, pellagra, scurvy, vitamin C, and many more diseases are given. I wanted to go through them, and it is really very important part of your exam. So you may probably expect few questions from these non-communicable diseases. And next, these important days, remember in your mind calendar, fix these days: and anti-leprosy day 30th January, World TB day 24th March, World Health Day 7th April, Malaria Day 20th August, and Diabetes Day 14th November, World AIDS Day 1st December. That's it. So thank you. Whatever doubts, any questions, you can always contact me. All the best for your exam.